I'm Shanna Henk, and I'm going to tell you about cryo-electron microscopy, or cryo-EM. Cryo-EM is a type of microscopy, so first I want to start by giving you a really quick crash course in microscopy. Light microscopes can only be used to visualize structures that are larger than the wavelength of light. To see much smaller structures, you can use electron microscopes because the wavelength of electrons is much smaller. Transmission electron microscopy, or TEM, is a type of electron microscopy in which electrons are directed through a thin section of the material to be visualized. It's a higher resolution than scanning EM, or SEM, which is used to visualize the surface of 3D objects because it detects electrons that have bounced off the surface. You can see the difference in the rectangle at the bottom right of the slide. In cryo-EM, you can see the sample in its native environment and conformation, since it's not fixed or stained in any way. Instead, the sample is frozen very rapidly. Formation of vitreous or amorphous ice rather than crystalline ice is important because water crystals can interfere with the ability to see the object. Electrons are shot through lenses that focus the beam through the frozen sample and onto an image recording system composed of a fluorescent screen and digital camera. The camera captures the electrons to form a 2D image. An algorithm builds a composite 3D image of tens to hundreds of thousands of images from different angles. In 2017, the Nobel Prize in Chemistry was awarded jointly to Jacques Dubochet, Joachim Frank, and Richard Henderson for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. This technique fits into Module 4 because it's a way to visualize and understand proteins. It allows understanding of the structure of the protein, which can help understand how proteins interact or function. Advantages to cryo-EM include the rapid freeze of the structures that maintains their shape in their native conformation. Only small amounts of sample are needed, and it does not require protein to be crystallized. Also, sample prep is supposed to be fairly easy. Disadvantages are largely that the equipment for doing electron microscopy is very costly, and according to my mother-in-law, who was a microscopist while she was working, sample prep can actually be kind of finicky. While I already compared the technique to standard TEM and SEM, I'd now like to compare it to two other techniques for examining the structure of samples such as proteins. The first is X-ray crystallography. Protein molecules or other molecules are crystallized. An X-ray passes through the crystal, producing a diffraction pattern. Algorithms are used to determine the location of each atom in the protein from the diffraction pattern. It works on any size macromolecules. A drawback is that some proteins cannot be or are hard to crystallize. Cryo-EM works on those proteins, however. Crystallization can sometimes cause unnatural conformation changes that are thus functionally irrelevant. Another method is nuclear magnetic resonance spectroscopy, or NMR. For this technique, samples must be dissolved and radio-labeled. This method develops a picture of a protein based on the way the protein's atoms interact with a magnet, its resonance frequencies. The structures are closer to the real protein structure than with X-ray crystallography, but it only works on small molecules. In cryo-EM, the sample is frozen in its native state, so there is no weird conformation change And also there's no radiation damage to the structure, which is common with other types of electron microscopy because there's such a high level of radiation involved with the microscopy itself. It can be done on any size macromolecule, and it provides near atomic resolution and fast sample prep. Controls are not used in cryo-EM in the same sense as in other techniques we've studied in this class. There are no positive or negative controls for this technique. When controls are used, they are likely to be study specific. For example, looking at a protein before and after a cell is infected with a virus. With cryo-EM, it's more a matter of optimizing the sample preparation conditions for the best visualization. Next, I'd like to show some cryo-EM figures from the primary literature. This first figure is from a paper that applied single particle cryo-EM using a new electron source, energy filter, and camera to give better resolution than had been previously seen. They achieved a 1.22 angstrom resolution, giving an atomic resolution reconstruction of a protein molecule. The protein they examined is a human membrane protein known as GABA-AR. The figure shows the top and side views of the GABA-AR molecule. It's a homopentamer, or a molecule with 5 identical subunits that you can see clearly in panel A. I think it's a pretty dang cool looking protein reconstruction. The second figure shows 3 different representations of the structure of the human cohesin NIPBL DNA complex. The first is a schematic, the second shows the surface of the complex, and the last is a cartoon of the complex from the opposite side of the structure. Subunits are colored the same in each panel. Cohesin is a complex of proteins that organizes the eukaryotic genome by extruding DNA loops. It also mediates sister chromatid cohesion by trapping DNA. To examine how it does these tasks, the authors determined the structure of the cohesin complex bound to the DNA loader NIPBL and DNA. NIPBL is shown in pink and DNA is in gray, and everything else is part of the cohesin complex. Cohesin and NIPBL form a tunnel that traps the DNA. The structure also engages cohesin's ATPase head domains and promotes ATP binding. In the third study, B-cell antibodies from cows vaccinated against foot and mouth disease virus, or FMDV, were tested against 2 serotypes of FMD. The virus is icosahedral, and the first 2 columns show the axes of the shape and the 3D structure. In the second column, the colors show the depth of the structure, so that red areas stick out and blue are more central. The authors identified 2 antigenic sites on the virus, which were bound by 2 broadly neutralizing antibodies, W125, which is the top row, and W2, which is the bottom row. In the third column, the pink areas show where the 2 antibodies bind to the antigen sites. The figures in this talk were from these 3 papers, respectively. And these are the sources that I used in developing this talk. Thank you.